Sorry guys, I was trying to do too much this morning. Thanks for taking my scarf off. So here we are, lecture 6. Okay, so quick business. The quiz opens this afternoon. Don't forget to take it. I'm going to try to remind you guys as much as I can on Fridays until you get into the habit. Quiz every weekend till Friday. If you miss enough of them, it's bad. That hurts your grade. Recitation materials will go up today, so keep an eye out for next week's recitation materials. Exam 1 is coming right up, isn't it? Yeah. I have a lot of exams, so you can feel free to mess up on one of them at least. So it's going to feel like they keep coming up, and I'm sorry for that anxiety that produces, but you should feel good about it. So as you'll notice, and I feel like no one's listening to me and people are talking. Who's talking about the Ravens? Because that's over. Just talk about the Orioles, because that's not happening. No. Okay, good. Because I don't care about sports. I like the Tour de France, but now like Lance Armstrong has really messed things up. So I don't even know if I like that anymore. Golf is really boring. Cricket? I don't understand it. There's a wicket? I don't know. Okay. Anyways, cricket I really want to understand. So anyone who wants to school me in cricket, please come see me. Okay. Exam 1. It's coming right up. It's a recitation. So you'll see on the recitation schedule, it's where it says exam 1. That's not a recitation exam. It is the exam. If it's on the normal syllabus, because I forgot to take it off, I apologize. We're just going to have more time to learn things and lecture. And you'll also have more time to take your hour-long exam in your 2-hour recitation period. So the way I see it, it's all win-win-win in so many ways, because it should take some of the pressure off of trying to get an exam done in this crowded environment, in this distracting space with no time at all. And then some of you might be like, well, it's not fair on Monday. Well, you know what? You get it out of the way first, and the other people have to deal with new material that I'm teaching. So there's pros and cons to everything. So, you know. Okay. Start thinking about the oral exam option. Anyone who's like, I want to try it, I will I will put out the schedule somehow next week, and we can start scheduling that. It will be on the same day that your normal exam will be, so it's not a way to get more time or anything like that. It's really, I'm guessing no one's going to do it this time. You're all going to feel out the exam and then decide when you're failing that you want to try that. But just putting it out there, it's going to be an option. And then start thinking about multiple choice questions, because starting next week, I'll put out a call for those. And if you submit a multiple choice question to me that is not recall, has no all of the above or none of the above answers, and is actually analytical, and I use it, you will get extra credit. You'll get an extra, what did I say? I forget. But it's an extra 100 points on your exam. It's whatever's in that, whatever I said to you in the past. Okay. So, where were we? What is that? Is that normally like little ears sticking out there? Isn't that funny? Right there. Those are modified pistons. I have them put up so we can understand all about this one. Okay, so where were we? So whenever I do this kind of review slide in the beginning of lecture, think of that as your study guide. Look back to these when you start to study for your exam and write down, because those are what I thought the main big picture points were of the last lecture. So if you're ever feeling lost and like drowning in details, go back to this and be like, okay, do I understand these concepts? Okay, I'm good. Okay, so we were talking about chromatin structure and function. That's you. We were talking about and that's basically the openness or closedness of DNA and how that relates to cellular processes that are happening, such as gene expression or mitosis. Those are just two examples of cellular processes where chromatin structure and function is really important. It tends to change. We talked about how histones bind to DNA. We spoke about the concept of post-translational modifications and their role in regulating protein-protein or protein-DNA interactions and the subsequent effect on cellular functions. That's a big, big picture thing to think about and make sure you understand it. All these little details I'm giving you, try to fit them into that idea. And then I started to introduce the concept of proteins having functional domains, and that's why I have Legos, and we'll talk about that shortly. And so I started by introducing the domains that we care the most about in this class: chromodomains, which bind to methylated lysine, bromodomains, which bind to acetylated lysine, and SH2 domains, which more I care about in signaling, but still I'm telling you about, bind to phosphotyrosine. Yes?
You want us to know how they bind and why they bind?
No mechanism, just the fact that they do. Yes. Okay. So, why? It's weird. I feel like those little things were not there before.
The screen is higher up this time.
Yeah.
The projection.
Can that go down a little? I don't know. Okay, whatever. We're just gonna go with it, guys. Gotta be flexible in life. Okay, so what do I mean by protein domain? Right, because people are always having a really hard time, and I could tell from exam, the questions and the answers on exams, people think of domains as a whole protein or as part of the DNA, or people think of it as the weirdest thing. So I want to drive this home. And some of you are like, this is so easy, why is she harping on it? Sorry, guys. It's not easy for everyone. Okay. So think of proteins as modular structures. And here, let's pull out some Legos. We're going to talk about this. So up on this, I have 1, 2, 3, 4 different proteins, and they have different domains that are highlighted. You know, they have little shapes that they put. And that just means here's your whole length of your protein, and at the C-terminus, there's 2 bromodomains on this one. Over here, there's a PhD domain, a bromodomain, a PWWP domain. There's millions of domains. Don't ever memorize them, because that's really dumb. But there's a lot of information you can get from domains. Who's taking 410? Anyone? Some of you guys are taking 410. And you guys had to look, with your short proteins, you had to look to see if there were conserved domains, right? So you can relate that a little bit to that, where if you have a conserved domain, and a domain is a functional unit of a protein, and it's conserved throughout many species, in the case of the project they're doing in 410, many species of bacteria, then you can say, well, this is probably this bit of the protein probably has an important function, and it's probably a real protein if it contains a conserved domain. So domains are functional units of a protein that they do something, they're functional. So for example, this helicase C domain is likely to do what? Have what kind of activity? Helicase activity, I know, really wasn't rocket science there. And so bromodomains, I already told you, they bind to what? Acetylated lysines. So this protein here is likely to be involved in what kind of general function? Well, acetylated lysines on let's say we're talking about acetylated lysines on histones. And if this protein tends to want to bind to histones with acetylated lysines, it probably is involved with chromatin remodeling or gene expression or something having to do with something that histones are involved with. We're going real general here. We can get more specific, because what you'll learn about is how acetylation is involved in activation of gene expression, and then when you start to see a protein with a bromodomain, you can start to think that maybe it has some function in activating gene expression because it binds to acetylated lysines on histones. Okay, so functional units of a protein. So what am I talking about? Okay, so we're going to use these. Okay, so I always say to students that proteins are modular, and I use all these crazy examples of what modular means. Who knows what modular means? What's a off the cuff definition of modular? You could think of modulars, like when you buy, but you guys aren't in construction, so you wouldn't be thinking about modulars. Yes.
It's like a sub-unit in case, like you buy multiple bigger unit.
Right, so a subunit. You can combine multiple subunits together to make a larger unit, or a larger complex that may have some function. So now let's start thinking evolutionarily, okay? Who was it someone said science only makes sense when you think about it in the context of evolution? Someone said that. So if you think evolutionarily, if you were like the first, and now this sometimes can be challenging for people, so we're going to break it down. So if you're like the first species that ever existed, and through all this mutation and whatever, you evolved some domain, some protein that has a part that's really good at unwinding DNA, right? That it's really good at it, and it makes that species survive. And so that little bit of that protein is going to kind of be passed along as the species evolves into more complex species and new species and whatever, and we get to where we are today, right? And so if that little bit of that protein had helicase activity, such as this helicase C domain, and it survived through all this time, then you would think that just, it doesn't make sense for each organism to suddenly create some new functional domain of the protein when this one works just fine, right? I don't know if that makes any sense. You don't want to constantly, we don't want to reinvent the wheel again and again because that's kind of wasteful of energy, and so different species tend to keep the same little subunits of proteins that do certain functions. So let's say you have a protein. I'm going to make a protein out of these. This is cool because I haven't played with Legos in a while, so I want to thank you guys. Okay, so here's my protein, and let's pick some little things. Let's say this part, this green part here is, let's say this green part here is like my bromo domain. And this yellow part here is like some other, I don't know, some DNA binding domain. And another color, how about some red? Red. And this red part here, some kind of helicase domain. They're all different. So it's one protein with these different parts on it. And mind you, I made it pretty linear for the purpose of explanation. But you know, proteins are not usually linear unless you denature them. So you have this domain that does this, and then this domain does something else, and this domain does something else. It's still one protein, but each little functional unit has its own job. And that's what proteins are. They are basically a bunch of functional units. If you have a really small protein, it probably has only one domain that does one thing. These large proteins, this one protein that I work on has a ubiquitin ligase domain. It has a DNA binding domain. It has a transactivation domain, which activates gene expression. It has a nuclear localization signal. It has all these different parts to it that do different things. So sometimes when people are studying proteins and their function, they'll put it into a database, they'll blast the sequence, they'll find the different conserved domains, which might come up as these different kind of things that are up here. And if you want to study the function of your protein, it's some unknown protein, but you found by similarity it has these different domains, what can you do? Let's say this is your bromo domain, and here's your helicase domain. what can you do to your protein to question whether or not it has a true bromodomain? Yes?
Introducing acetylated lysine.
Well, it binds to acetyl. So you'll have, yes, so you can assess whether it binds acetylated lysine, right? That's pretty much what you're trying to get at, right? Because if this is your bromodomain, this part of your protein is going to bind to your lysine on your histone. That's been acetylated. So let's say you want to definitively show that that is the case, and you're kind of now going on. We're going to start getting into our recitation stuff for next week with this question. So what can you do? You can remove any of these domains, right? You know the sequence of your protein. You've blasted it. You've pulled out different. You've blasted it. You know where the different domains lie in your protein. You know this is your bromodomain. So if it's a true bromodomain and I take it off, should it bind to acetylated lysine? No. Should it activate gene expression or whatever I think it's supposed to do? No. Right? Let's say this is a helicase domain. What would I do if I want to show that it affects, that it has helicase function? Take it off and see, does it still unwind DNA? Do I still get whatever function I think, probably DNA unwinding or having some sort of transcription or replication or something like that, right? See if I still get that. So this is how science is done. We'll chop up proteins and see if they still do what you think they do. Yes?
I was going to ask, multiple of the same domains, do they all have the same function, or do they do different things if they...
So like where there's two bromodomains? So the question is, if there are multiple of the same domains, do they have the same function or do they do different things? So two bromodomains probably means that it binds to two acetylated lysines. And we're going to get to, as we talk about the histone code, we'll start to think about that a little bit. Okay. Where did I have? If I lose this, someone's going to get upset. Okay. So this is just a figure here of the bromodomain of GCN 5-base of protein. Don't memorize it. That is binding to acetylated lysine. So remember, a bromodomain binds to acetylated lysine. It doesn't have an acetylated lysine. It doesn't acetylate lysines. It is not an acetylated lysine. It binds to acetylated lysines. Okay, so let's see if we can really make it sure everyone gets this. Okay, so what does all this domain and post-translational modification talk have to do with chromatin structure and function? Right? I just went on and on about domains. I was talking about chromatin structure and function. Where are we? What the hell does this have anything to do with? What I'm trying to get you to understand is that histone modification affects chromatin structure and function through increasing DNA accessibility or recruiting proteins that are going to affect the structure and function. Okay? So this is just showing your histone with your DNA wrapped around it, and it's showing all the different types of modifications you can get. And this is mostly just on the core histone, not the histone tail sticking out. And so you can see the blue are methylation, the yellow are, so blue methylation, yellow acetylation, red phosphorylation. We've got some pinkish, purplish, ubiquitination, and then some places you'll get ubiquitination or acetylation, because those tend to compete with each other, and actually also with methylation. So I just want you to understand, you don't need to know where each thing is modified, that's not useful, but to understand that there's a lot of modifications happening, and because this changes the shape of the histone and creates or removes binding sites for other proteins or affects interaction with the DNA, this is how chromatin structure and function is regulated. So you're either opening or closing the DNA to allow for gene expression to happen or not. That's the simplest explanation of it. Questions about this major concept? Huge concept. You will not understand the rest of the class if you don't understand these kind of things. Okay. Everyone good? I know some people say to me actually in my reviews that I spend too long on this part, but I'm guessing those are like the 5 people who came in understanding it. So I'm hoping that harping on it will make it clear. Okay, so Histone acetylation is associated with active chromatin. Active chromatin means open chromatin, most likely involved in active gene expression. So you've got your closed chromatin here, and your histone acetyltransferase, your HAT, will come along and acetylate some lysine, and that will result in opening up the chromatin, so transcription factors can bind and RNA polymerase can come in, and you can have gene expression. There's another enzyme. Don't forget, these modifications are all reversible, right? Because if you have this permanent acetylation, well, that wouldn't be good, would it? So then there's a histone deacetylase, and that's involved in removal of the acetyl groups, right? It's right in the enzyme name, so you guys shouldn't have too much trouble remembering that. Deacetylation, right? Histone deacetylase. So that's a HDAC, and that removes the acetyl group and allows for closing up of the chromatin. Any questions about this? Okay, so acetylation, what's it really doing, right? So I'm telling you it's associated with gene expression, it's on a lysine, it binds to bromodomains, but what does acetylation really do? Well, it does recruit bromodomain containing proteins, but the other thing that happens is it also opens up the DNA by altering the charge on the lysine. So lysine is a positively charged amino acid. What's the charge of DNA? Negative, so that's tight binding, right? But if you acetylate your lysine on your histone, you're going to remove that positive charge. That's going to help to open up the DNA. So that's what's really happening there. Right, so you get acetylation by histone acetyltransferases and deacetylation by HDAC. Yes?
The acetyls are adding a negative charge to it?
The acetyl group itself isn't adding
Because
it's
neutral,
just neutralizing it, right?
wouldn't that still attract?
We will not have the tight binding, right? Positive negative is going to be tight binding, and if you neutralize it, the acetyl group, okay, who is a chemistry person? Is the acetyl group negatively charged? No, I think it just neutralizes it. Come on, chemistry people. I know someone's in here.
All I know is that if you remove a charge, a neutral charge is still going to attract a negative charge.
But it's not going to be that tight binding. Because really the purpose, that's not the only thing that's happening here, right? You have the loosening of the DNA that's wrapped around your histone, and you're also going to have these other modifications that are going to come and recruit chromatin remodeling complexes that are going to help to slide things away. So it's not just acetylate open. It's not that simple. Yeah. It loosens it up. Okay. Okay, so there's lots of enzymes that are involved in histone modification. These are some of them. Do you not memorize these? Hopefully you guys are understanding at this point that I do not want you memorizing things. So there's histone acetyltransferases that acetylate, histone deacetylases that remove acetyl group. Then there's histone methyltransferases. What do you think histone methyltransferases? Transfer what? Methyl. Yeah, methyl. Yeah, they methyl. Right. Okay. So histone acetylation also creates binding sites for proteins that contain bromodomains. So I talked about how acetylated lysines will bind. Bromodomains bind to acetylated lysines. So these bromodomain-containing proteins will come. They'll bind to the acetylated lysine on your histone tail, and they'll recruit other proteins that will do something. And a very common one that will be recruited might be a nucleosome remodeling complex that's going to come along and slide the nucleosome out of the way to expose a promoter or something like that. There are also other proteins that could be recruited, like more histone acetyltransferases. If you want more acetylation, you could be recruiting demethylases if you're trying to remove methylation. A lot of things like that. Oops. Okay, so that's acetylation, right? Now we've got methylation. So histone tail methylation creates binding sites for recruitment of chromodomain containing proteins. We talked about chromodomains. These are chromodomains, just different domains that tend to bind to methylated lysines. And they act the same way. So in this figure, you can see you've got your nucleosome, you get methylation. That will result in recruitment of chromodomain containing proteins, which will then recruit some other protein complex that's involved in something. It might be nucleosome remodeling complex that's helping to close up the DNA, because methylation is often involved with silencing. So methylation is involved with heterochromatin formation. So that's that tight packed DNA that's associated with no gene expression. Oh man, I just answered my own question. What is the likely effect on expression of genes located in regions bound by histones with methylated tails? For those who are not paying attention.
Transcription.
The transcription, right? Because I said it's involved in heterochromatin formation. Yes.
So what would be the difference between that and acetylation, since they both have to do with DNA being lost?
So the difference is with acetylation, you're opening up the DNA, which is allowing transcription to happen. And methylation is going to recruit different protein complexes that are going to be more involved with closing up and making a higher order compacted condensed DNA. Other questions? Did I answer your question? Okay, so enzymes that methylate, histone methyltransferases, and enzymes that demethylate, histone demethylases. Pretty clear, right? Right in the name of the enzyme. It's nice when enzymes kind of are named by what they do, because then you don't need to remember them. It's right there. Okay, so chromodomains are typically found in proteins. This is obvious. I shouldn't even have to say it, right? Chromodomains are typically found in proteins that mediate transcriptional silencing. So I said it's involved with silencing. It's involved in tighter packing of DNA and heterochromatin formation. So it's logical that chromodomains would be found in proteins that mediate transcriptional silencing. I wish I had some sort of food right now. Who's hungry? Yeah. You should start bringing donuts to class. Okay.
So getting.
There we go. Oh, wow, you are ready.
Jeez.
That is good stuff.
If anybody's interested in helping disabled students in Towson, so getting.
I'll help disabled students. Okay. Okay. And it's not just about the candy. It's not just about the candy. Okay. Lysine acetylation. Are you good? Okay. Lysine acetylation and methylation are competing reactions. So I told you a lot of things happen on lysines. You must have thought about this, right? Wait a minute, this is all happening on lysines. Oh my god, how many lysines are in a protein? So these are competing reactions, which makes some sense actually, right? Because acetylation and methylation at the same time, it does happen at the same time, but that can become confusing because one's involved in activation and one's involved in repression. Now that's a very simplistic way of looking at it. In reality, methylation under certain conditions can be involved in activation, but I'm not going to bother you with that. Nothing's ever definitive in science. I want you guys to understand that. And that's really frustrating for a lot of people because you want things to just be black and white. It's either this or it's this and that's it. But that's not the case. So you can have, so here's your lysine, right? And you can get acetylated lysine, or you can get monomethyl, dimethyl, or trimethyl in your lysine. And there are different proteins that are going to recognize these different modifications. Question? Yes?
Do these cause different gene expressions, or is it still like the original just methylation?
Do they cause different gene expression? So this is really talking more about the overall chromatin structure. The specific gene expression is more regulated by different activators, different transcription factors and activating proteins that are going to be recruited to the DNA. Yeah. But this is very important in whether or not the promoter is even going to be exposed, right? Yes?
Do mono, di, or trimethylglycines have a greater effect on methylation?
You mean on like heterochromatin formation?
Right, and one have a stronger effect on that.
I know that. So the question was, well, whether or not you're mono, di, or trimethylated, will that have a stronger effect on the function? So I do know that different proteins do tend to prefer different forms of methylation. So it's a level of regulation. Whether it has to do with the intensity of the effect, I'm not sure. Yes.
I'm just wondering if whether it's like mono, di, or tri, how it's methylated, is that correlated with how the structure is formed?
Yes, so it absolutely does, because one methyl group versus 3 methyl groups is really going to change the shape of the protein, right? And that's going to affect recruitment of different proteins, right? Because don't forget, protein-protein interactions are all about interactions between the different side chains of amino acids, right? So if you have just something like one methyl group added, it's going to recruit a different protein than if 3 methyl groups are added. But I can't tell you whether or not you're going to have the higher order heterochromatin, like really tightly packed based on mono or tri. Maybe we should look that up. Let's make that an extra credit, okay? Because someone stumped me. A few people here just stumped me. So for those of you paying attention, you have 24 hours. Yes, I know it's Friday, tomorrow's Saturday, but I will check my email. So by, we'll give you till noon tomorrow. You need to let me know if monomethyl versus di versus trimethylglycine has any effect on intensity of silencing. How about that? I already know it is involved with recruitment of different proteins, but whether or not those proteins directly relate to silencing or structure of the chromatin. Yeah.
Putting aside the extent of regulation, as long as we have something methylated, it does have the effect, correct?
If you have methylation, then some protein will be recruited.
Okay.
How big is that? And most of the time, those proteins are associated with transcriptional silencing. Yeah. Okay. Okay, and so here's your phosphorylation. Don't forget the one I'm not paying too much attention to, but it is also very much involved in recruitment of proteins, is phosphorylation. And serines tend to be phosphorylated. So that's just addition of a phosphate group to the serine. And that also will be changing the shape of a protein and will result in recruitment of other proteins. That will do something. I'm being vague because there's so many proteins, right? And they all do different things. So I can't give you definitive like this does this. Yes? Yep, so that's SH2 domains. SH2 domains tend to bind actually, no. Phosphotyrosine. I think SH3 domain. But don't worry about it. The big ones for me are chromo and bromo. Okay, so going on and on about post-translational modifications. And so to understand really what the whole point of all this is really it's about so I want you to understand it's not one post-translational modification that happens, right? They happen in clusters, they happen in groups, there's a lot that happens. So here's your histone subunits, right? And you can have modification on the core histone, so that globular domain of your histone, or you can have it on these protruding histone tails, right? And look, you've got phosphorylation and then acetylation at multiple sites. You've got methylation and acetylation happening next to each other. And here, this does this show, that shows some ubiquitination, doesn't it? Somewhere, maybe. Yep. You can get some ubiquitination. So they're all involved in different things. So this takes us to the major concept of the histone code, which I guess we'll get to in a minute. Okay, so there are additional post-translational modifications that I did not talk about, but I want you guys to just understand. We briefly spoke about phosphorylation, ubiquitination. Also, there is sumylation, which is ubiquitin-like protein. And it all can happen on histone tails, kind of like in a big mess, like what you see here. And it bless you, this regulates different processes by recruiting different proteins that are going to do different things. Very basic concept, but important concept. Phosphorylation is involved in expression of genes involved in cell growth, as well as in chromosome condensation. Ubiquitination and sumylation are associated with transcription and with DNA repair. Okay, so here's the concept of the histone code. I just showed you these histone tails are modified like crazy, right? That's the histone code right there. That modification, in the whatever orientation the modifications happen, means something, okay? Because it creates a protein with a very different landscape, right? Very different, it has multiple binding sites that's going to recruit some other kind of proteins. So you can think of it as, so what will happen is you'll have recruitment of some complex of proteins, which the book refers to, and Alberts refers to as a code reader complex. So it's a complex of proteins that's going to bind to certain modifications of histone tails. And the purpose of it is to recruit other proteins that are going to affect chromatin structure and function. So that's the concept of the histone code. It's an important one to understand because so many different things need to happen with histones because gene expression and DNA repair and chromatin structure in terms of whether you're heterochromatin or euchromatin or whether you're condensing your chromosomes before mitosis. All these things are fairly complex and need to happen in a regulated fashion. So it would make sense that there would be some sort of way of relaying the message to the proteins that need to be recruited to do the job that needs to get done. Okay, do people understand? People often have a hard time understanding histone code.
So it serves to recruit the proteins that are needed for the process?
Yep, serves to recruit the proteins that are needed for the process. But it's a histone code because there's all these different modifications that happen, and each modification in different combinations could result in recruitment of a whole different complex of proteins to do a whole different function. Are we good with that concept? Yeah. I see some nods. Anyone know? Okay, so there are different outcomes for different modifications. Here are some examples. So you can just have something as simple as some methylation, and that will be heterochromatin formation and gene silence. So it will recruit. What kind of proteins do you think might be recruited? If you just have a little bit of methylation. Maybe something involved with a repressor, maybe. But really, chromatin remodeling complexes that are going to close things up and help to bind the histones to each other and create a higher order compact chromatin. We have repressors too, probably. Then you can sometimes have some methylation and acetylation on specific notice. These are so the 9 refers to the amino acid number of that histone tail. So if you have methylation on lysine 4 and acetylation on lysine 9 of one histone, I think H3, that will result in gene expression. If you have phosphorylation on serine 10 and acetylation on lysine 14, that might result in a different kind of gene expression. If you have methylation on lysine 27, that will and be involved with different functions. So how does this all work, right? How does this work? What's happening when you have these modifications? Specific genes are being turned on and off. Not really. So what's resulting in this specific genes being turned on and off or whatever? What's not resulting? What's causing that? Chromatin is changing shape, right? How is the chromatin changing shape? Is it just because you have modifications? Now, what are these modifications doing? Recruiting proteins. I hear lots of whispering in the front. If there's whispering in the back, I don't know because you're far away. I'll just believe there's whispering all over the place. Okay, so this is the concept of the histone code. Different modifications result in recruitment of different proteins, which result in different functions. Yeah.
Since methylation and acylation kind of have the opposite voting versus opposing, why would we get gene expression? Like, would we get it in this case, but maybe not if it was on a different amino acid?
Right, so that's why I said before, methylation is mostly associated with silencing, but not 100% of the time. So that's the thing to keep in mind with science, is that it's not always 100% of the time, but because I'm telling you methylation is mostly associated with silencing, I think I give you permission to believe that. But also keep your mind open to the fact that sometimes you get methylation with acetylation and it does result in gene expression. So there is some flexibility in that. Yeah. I do not know that acetylation is ever involved in silencing. But you know what? Someone might find that tomorrow. Any other questions? Yes.
So if these are on the tails of repeating subunits, where does the specific specificity come from? Like which leucine gets methylated or acetylated?
Okay, so which lysine would get acetylated? So you've got, so are you saying, okay, are you asking how does the hat know to acetylate a specific lysine? It has to do with how the hat is recruited and what, what lysine is most in the nearest proximity to the active site of the hat. And that is regulated by other proteins. So what we'll talk about is that you'll have some sort of maybe a general acetylation that will happen, which will recruit some bromodomain containing proteins. That acts as a linker to recruit other proteins. So that might recruit another protein that has hat activity that's going to acetylate somewhere else. And it's the recruitment of the different proteins and the histone code that helps to have this very specific modification and then recruitment of proteins that are going to affect the structure and functions. So as someone who's worked on ubiquitin, I can tell you that there could be 50 lysines in a protein, right? And people always want to know what ubiquitin is being or what lysine is being ubiquitinated. And so if you mutate your lysine, let's say you change all your lysines to alanine, so it can't be ubiquitinated anymore, you're still going to get ubiquitination, you're still going to get degradation until every last lysine is gone, because there's some sort of flexibility. It's crazy, I know. That's a different story. Okay, any other questions? That was a good one, though. Yeah. Other questions? Okay. So histone code readers might recruit other writers that will help to spread the modification along the chromosome. So here's how it might work. So you'll have your nucleosomes, right? And some gene regulatory protein will bind here and recruit some other protein, which will then modify the histone. Okay? And so it all has to do with what's recruited to where and the proximity to what lysine is exposed. And then that will recruit, so you have this modification, right? So let's say this is acetylation. No, let's call it methylation because we're going to try to silence things. So then you'll get methylation here, and that methylation will be recognized by another protein that will come along, and it's reading the histone code, so called our reader protein. And it will also act to recruit another protein, which might be another writer protein, which will position a hat, right, this writer protein, within close proximity to the next histone, and that will allow for methylation of that next histone. And so on until you get this methylation across a stretch of chromatin to help to silence that region. Okay? So it's all about recognition of modifications and the recruitment of other proteins, these other things, all in some end goal. Yes?
So during gene suppression, is it likely that there isn't just the nucleosome, but a composite protein around the nucleosome?
Oh yeah. So with repression, is it just the nucleosome or is there a complex of proteins around the nucleosome? Yes, there are many proteins involved with repression. You'll have methylation, and that methylation frequently recruits proteins involved in repression. Yeah, absolutely. Okay, so I guess we're kind of done. Yeah, so we'll finish this later. I will see a lot of you in about 10 minutes.
Great.